Since the last nugget on how animal testing kills humans, I've had several inquiries about what viable alternatives there are. By the way, now that Ubi's back, you'll be hearing this in the background. Now, animal testing isn't just dangerous for humans, completely ineffective in producing any scientifically viable result, and totally driven by greed, but it's also not the only option. During a government meeting about funding for research, former U.S. National Institutes of Health Director Dr. Elias Zerhouni admitted that experimenting on animals to help humans has been a major failure. He told his colleagues, "We have moved away from studying human diseases in humans. We all drank the Kool-Aid on that one, me included. The problem is that animal testing hasn't worked, and it's time we stop dancing around the problem. We need to refocus and adapt new methodologies for use in humans to understand disease biology in humans." Stated most simply by Drs. McLennan and Amos, there is no doubt that the best test species for man is man. So what are the alternatives? I'll be pulling a lot of my information from the Johns Hopkins University Center for Alternatives to Animal Testing. Now, we don't have time to cover all of the alternatives, but here are the basics. There are three main categories. The first is sophisticated tests using human cells and tissues. This is known as in vitro methods. Then there are advanced computer modeling techniques, often referred to as in silico methods. And third, there are studies with human volunteers. These and other non-animal tests are not hindered by the species differences that make applying animal test results to humans difficult or impossible. And they usually take less time and money to complete. So here are some examples of in vitro testing. Harvard University's Weiss Institute of Biologically Inspired Engineering has created organs on chips that contain human cells grown in a state-of-the-art system to mimic the structure and function of human organs and organ systems. The chips can be used instead of animals and have been shown to replicate human physiology, diseases, and drug responses much more accurately than crude animal experiments do. Artery on a chip has been used to study heart disease. Canadian scientists have developed microfluidics chips to enable fragile blood vessels to be studied easily and cheaply. The device will allow thousands of potential new drugs to be routinely screened for their effects on blood vessels. Harvard's Weiss Institute produced a breathing lung on a microchip in 2010. Weiss researchers were later awarded a grant to develop a spleen on a chip to treat sepsis. Now they have produced a physiologically realistic gut on a chip, which enables them to study much more complex interactions than previous cell cultures or animal models. They are currently working on more than 10 different organs, with the ultimate goal of linking them together with fluidic channels to produce a more representative model for testing medicines. Cetox, bought by Ciprotech, developed a method to assess the potential of a substance to cause a skin allergy in humans that incorporates MatTech's epiderm tissue model, a three-dimensional human cell-derived skin model that replicates key traits of normal human skin. Researchers at the European Union Reference Library for Alternatives to Animal Testing developed 5 different tests that use human blood cells to detect contaminants and drugs that cause a potentially dangerous fever response when they enter the body. The non-animal methods replace the cruel use of rabbits in these painful tests. Now some examples of in silico models. Researchers have developed a wide range of sophisticated computer models that simulate human biology and the progression of developing diseases. Studies show that these models can accurately predict the way that new drugs will react in the human body. And now some examples of research with human volunteers. A method called microdosing can provide vital information on the safety of an experimental drug. Volunteers are given an extremely small one-time drug dose, and sophisticated imaging techniques are used to monitor how the drug behaves in the body. Advanced brain imaging and recording techniques, such as functional magnetic resonance imaging with human volunteers, can be used to replace archaic experiments in which rats, cats, and monkeys have their brains damaged. These modern techniques allow the human brain to be safely studied down to the level of a single neuron, as in the case of intracranial electroencephalography. Yet another alternative are human patient simulators. These are strikingly lifelike computerized simulators that breathe, bleed, convulse, talk, and even die. They've been shown to teach students physiology and pharmacology better than crude exercises that involve cutting up animals. Most high-tech simulators mimic illnesses and injuries and give the appropriate biological response to medical interventions and injections of medication, and have been shown in numerous studies to impart life-saving skills better than courses that require students to cut into live pigs, goats, or dogs. So as you can see, there are plenty of viable alternatives to animal testing. The whole thing is a pretty shitty idea, regardless of the existence of alternatives. But given that there are alternatives that are cheaper, cruelty-free, and more effective, there is really no fucking reason to continue testing on animals. Please visit BiteSizeVegan.com and sign up for the newsletter, and be sure to subscribe to the channel, as well as watch all of the Bite Size Vegan nuggets on the Bite Size Vegan Nugget playlist. And if you're a Facebooker, check out Bite Size Vegan's Facebook page, and go ahead and friend me too to see what I'm up to. Plus, follow on Twitter at BiteSizeVegan.